Welcome to this exciting session. Gene editing accelerates first generation nears approval. Glad and not surprised to see it's a very well attended session, and I think it should be guaranteed to be interesting considering the subject matter and the quality of the panel. So I'm Tim Farries. I'm one of the principal consultants at Biopharma Excellence. I'm particularly interested in this field, of course, not just because we do a lot of consulting in this area, but actually you go back to when I was a product developer. It was in the early days of genome editing. Some of the early work I was somewhat peripherally involved in, but it was still nice to even have that much involvement in only genome editing, fruit flies and the like. But the panel here, four experts representing leading players in the field, driving the field forward, as put in the title of the session. It's going into the clinic, heading towards approval, and so many following in, and pursuing the same course. So I won't give you their full biographies. They're available online. But we have from the left, Fred Chereau, President and CEO of LogicBio. Albert Seymour, President and CSO of Homology Medicines. Mark Shearman, Executive Vice President and CSO of Editas Medicine. And Devyn Smith, CEO of Arbor Technologies. We do hope to allow some time for some audience questions later, so do try and think of your questions and get them ready for when that happens. We've got a whole hour, but I think that hour will disappear very quickly because there's so much I'm sure our panel will have to say. And I think if we start off by going through the panelists one by one, telling us what they're doing in the field, what's special and unique about the way they're doing it, the technology, and how they're approaching it. So start with you, Fred.
Sure. So good afternoon. So I'm Frédéric Chereau, the CEO of LogicBio. LogicBio is a genome editing company developing a couple of platforms. We have our first genome editing platform called GeneRide, which is harnessing a naturally occurring process or DNA repair process, which should help us to lead to a long-term protein expression. We also have a capsid engineering platform developing interesting capsids, for different tissues and different indications. And last but not least, we have developed a manufacturing process called Mavericks, overcoming, I think, one of the Achilles tendon of AAV gene therapy to date by delivering high potency and good quality products.
Thank you. Albert.
Yeah. Hi, everybody. Albert Seymour, President and CEO of Homology Medicines. Homology Medicines is a genetic medicine company, and we are developing off a platform of 15 novel HSCs that were discovered in human stem cells, and we call them AAV HSCs. And we can attack different genetic diseases with three components of our platform. The first is more of a traditional gene therapy. With these naturally occurring AAVs, we get distribution to a lot of different tissues and organs that are important in a variety of different disease states, so we can align that up. Secondly, we can take advantage of the fact that these AAVs and AAVs in general use a single-stranded genome, and so we can engineer the inside of that genome to integrate into the genome via a process of naturally occurring homologous recombination. And that's one where we're utilizing that to turn genes on. We have a lead program in the clinic right now in phase 1 for PKU using that approach. And then lastly, we can take advantage of the platform to express full IgG, full heavy and light chain IgGs for a disease called PNH. And so we can utilize the approach to sustain the expression of antibodies with our platform.
Hello, everyone. Mark Shearman. I'm the Chief Scientific Officer at Editas Medicine. So Editas is a CRISPR company. It was the first publicly traded CRISPR company in 2016. We have proprietary access to SP-Cas9 and AsCas12a nucleases. Currently, we have programs in the clinic in ophthalmology and hematology, and have a suite of programs in the immuno-oncology space as well. In addition to the CRISPR-type nuclease activity that I think people are familiar with, we've also developed a technology called SLEEK, which allows us in ex vivo cell preparations such as T and B cells, induce pluripotent stem cells to knock whole genes in. And so this is a very powerful technology for multiplex editing. Since 2016, in addition to advancing the therapeutic pipeline, we've also developed a number of proprietary in silico-based software programs. A lot of the focus now as gene editing gets to the clinic is on the ability to control on-target editing and have an awareness of and an estimate of the risk associated with the potential off-target editing. So we spent a lot of time developing those technologies and coupling the in silico work with detailed analytics.
Thank you. Devyn?
So Devyn Smith, Arbor Bio. So Arbor, we are in the CRISPR space as well, similar to Mark at Editas. We take a slightly different approach. We're looking broadly at all of the metagenomic wonders that have happened through evolution and have built a platform to really capitalize that and identify through the tens of thousands of potential editing approaches that have been developed evolutionarily to bring those in and select those that have the differentiated properties and qualities we want, so that we take a toolbox approach to the space so that we have a variety of editors that have a variety of PAMs, or the binding sites, as well as different sizes. We prioritize off-target profiles because we don't want that. We prioritize on-target and take those systems and put them in through a mammalian engineering platform where we can engineer them to get their levels up to therapeutic relevant levels of editing. We use those chassis, these nucleases, that then can be utilized as a therapeutic themselves or can be coupled to different approaches, whether it's a deaminase or a reverse transcriptase or even these CRISPR-associated transposase. That allows us to have breadth across the way to think about editing the genome, because it's clear that every disease in the genetic space requires a bespoke approach. I think that's something I think all of us have learned over the last 6 or 7 years. It's not as easy as, we've got one nuclease and we're going to cure every disease. I think it does require a fair amount of work, and folks like Editas and Homology and others have proven that. So.
Thank you all. And you're probably preaching to the converted here very much, but maybe you'd like to comment on what you can do with genome editing that you can't do with regular gene therapy. What doors does it open? Maybe Mark and then Albert?
Sure. So the principle of the CRISPR-based approaches are to make relatively small indels, so insertions, deletions. This can be iterated whether it's a single or a dual guide. And I think the advantage really is that it pretty much opens up a very large target space. It's very flexible. I think as I mentioned, you can also couple this with a knock-in as well as knockout strategy. And since we're modifying the DNA permanently, this is a very ultimately a convenient therapy for a patient to take, both in terms of compliance as well as having a sort of potentially a lifetime modification of their disease, or even in an ideal situation, a cure.
Thank you.
Yeah, and just to add on to that, Mark, I think when we think about diseases and focus areas of gene editing, I think you really want to go into, and it gives you the ability to permanently correct a cell at the genome level. So you can target the direct cause of different genetic mutations, whether you insert a gene that you want expressed or whether you knock out a gene that you don't want expressed. And I think the key thing there is it allows you to get into those cells that would be rapidly dividing. So for example, if you wanted to get into a very young child and have the liver that's going through many rounds of division, you could do that. Or if you wanted to get into hematopoietic stem cells and aspects like that, you almost are dependent on being able to go in and correct the cell at the DNA level in order to get that long-term durability over time. With gene therapy, you can do a lot of these things, but you can run into an element where if the cell's dividing or the tissue's dividing a lot, you can start to dilute that over time, depending on that cell. So if you go into a cell like a neuron that doesn't divide, gene therapy is great. But if you go into some of these more dividing cells, you do have to be cognizant about the a dilution of that effect over time. And in the absence right now of being able to redose with some of these delivery approaches, that really gives gene editing a very unique advantage over some of the gene therapy approaches.
Thank you. All very great answers. Anything to add to that? Well, perhaps we'd move on to something that's quite close to my heart, of course, is regulatory and ARMs. The FDA's brought out its draft guidance not very long ago, a few months ago, and ARM was quite involved in that. Devyn, what do you think of those? And how else can the FDA help to facilitate the clinical trials process?
Yeah, so as ARM has done a really nice job of, when this draft guidance came out, we actually got all the editing companies together, and we actually put forward a concerted view to the agency of this is what we as an industry feel is appropriate, what we like and what we don't like and what we'd want to see change. And I think having that concerted view by us as an industry, I think is critical for us being successful. So then the FDA is not getting 15 different approaches that each of us, think are slightly better, and it seeds confusion. So I think having that voice of ARM, being able to do that as a concerted group where, even though we have different technologies and different approaches, we all want to be successful, and one way to do that is we've got to help and partner with the FDA to get through these things.
Do you think that guidance is the end for a while, or there's a need for more guidance, more specifics, distinguishing in vivo, ex vivo?
I will take a crack, and it'd be interesting in others' view. I mean, honestly, I think we're at the early days, right? It's still early days for the agency. You look at least, where are the patients being treated in editing trials today? Most of them are actually being treated ex-US in the in vivo side. I think Editas is one of the few using CRISPR that actually has a trial ongoing in the US. So I think it's early days still for the FDA, and we need to continue to partner to help them understand the good, bad, and where the opportunities are, and how we can make sure we have safe, efficacious therapies.
Okay. Fred, how do you find the regulatory process?
Well, I think, does that work?
Yeah,
I think Devyn said it very well. First, I mean, we are at the early days of gene therapy, so even earlier days of gene editing or prime editing or base editing. Even if the science is going very, very fast, I think we need to continue to work with the agencies, and not only the FDA, but others, to continue to educate them and share the learnings to be able to provide the best possible guidance, which can guarantee the safety of the patients, but also the feasibility of drug development. And as Devyn said, I mean, it's interesting to see that very few gene editing company or in vivo gene editing companies have been doing it in the US. LogicBio has done it too, as well as Editas. And I don't know why, but I've always believed that it's a partnership between the regulatory authorities and the companies when you are developing so innovative therapies.
Okay. And events like this, and ARM is very good at bringing companies together. Yes, go for it, Mark.
So one thing that I've found is, obviously the FDA with any new technology wants to assess risk-benefit, and particularly the requirement for a proof of direct benefit. I think as we push new technologies, particularly into rare disease areas where there isn't much of a precedence, I think we're running into situations where the outcome measures are being transposed from known indications to new indications. And so I think the dialogue with the FDA as to how you interpret that information, or their openness to accept data from novel endpoints, will be important.
Okay, absolutely. And I was just going to say, one of the things about ARM is it brings developers like us together, helps contribute to a common viewpoint on this. Do you find, how do you find that helps? Do you think ARM helps bringing fellow developers together, Albert?
Yeah, absolutely. And I think to Devyn's point about the guidance, pulling everyone together and contributing to that. I think the other thing too is that there's so many different diseases out there that need technologies, and I think what ARM does is it brings together a lot of the companies that work on these technologies so that we can learn from one another as well. And I think we talked a little bit about working with the regulators. I think that's also another area. What have we learned that, in a sense, pre-competitive? Just how did you go about off-target analysis? How did you go about this? If there are things we can do together to help collectively the whole community, I think that's where, at least coming to the meeting at ARM and interacting with all the companies here has been beneficial.
Okay.
Well, and I would say, I mean, unfortunately, we faced some safety issues in our clinical trial, and I think it was very reassuring to see that other companies who have faced the same one have been willing to share a lot about their learnings and how they have been able to overcome this issue. And I think that's one of the very interesting part of not only ARM, but the regenerative medicine space today.
And on that safety issue, do you find that the field now has confidence in the technology? The technology, when I was first involved, we didn't have much confidence in. It was not specific, but 20 years later, it looks a completely different picture. Would you agree with that?
I'm not sure the picture is completely different, unfortunately, but I would say that I hope all the people here are very confident in what they are doing. We have made tremendous progress, but there is still a lot to do, as it has been said before, and I think one of the, it has been said in the previous panel, I think, but we should not mix up gene addition or gene therapy and gene editing. Gene editing has been the new shiny thing, or I forgot exactly how Josh called it, but it's still very early. It goes one step beyond to the DNA in some ways, and I think we have to be very humble with the science and continue to work step by step and continue to share the learnings.
Okay, anybody else?
I mean, I think I would echo what Fred said. I think it is early days, but I think it is, I think one of the reasons there's so much interest in editing is that, if you think about 20 years ago, when I first got out of grad school, to think that you could actually edit and completely fix a problem in the DNA, I mean, couldn't even dream of that, right? And we can do that today. And so I think the promise of editing is so amazing that I think that's why you see so much interest in it, because it brings our ability to treat disease, not to treat it, but actually to cure it. And I think, and do it in the actual endogenous DNA rather than having an ectopic chunk of DNA. I mean, that's the promise, and that's why I think there's so much interest in this space. And are we gonna get there tomorrow? No, it's a journey, but you're seeing a lot of great progress. And that's what I find completely exciting about this space.
And while I'm with you, Devyn, the investors, do they understand it? Do they have the same expectations, or would you say their expectations are different from you as a developer?
Do you want me to start? You're looking at me, so. I think that, I think investors understand the promise. So, last November we raised $215 million in our Series B, one of the largest Bs last year. Why? Because I think investors understand what the promise in this space looks like. You look at the public companies, and Mark can comment on that, but they continue, the valuations have held up remarkably well considering the, what's happened in the market, unfortunately, over the last year and a half or 2 years. And so I think investors get it, but I think it's incumbent on all of us in this space to continue to execute and hopefully generate that positive clinical data so that momentum continues.
Yeah, I mean, my experience is that the investors and analysts have been generally, their expectations of what are in line with what I would have imagined. I think with any new technology, obviously the focus is on clinical data and proving that it's safe and efficacious. I think typically they also want to see a projection of where the program's going, the timeline over which you'll be able to submit an application. And then I think they're also interested in, are you in control of the technology? Do you have all of the infrastructure to monitor the technology and to be confident that you can advance it? And then I think lastly, particularly in the cell therapy space, they want to be convinced that you can actually manufacture the product, again, in a controlled fashion. And then lastly, if you have examples where your process and your analytics have been reviewed by a regulatory authority, whether it's the FDA or other, that's obviously a confidence booster if it's been approved or accepted, I should say.
Okay, thank you. And Fred, you obviously deal with investors.
For a few more weeks, I hope, yes. No, more seriously. I think Devyn said it well. It's everybody sees the promises, everybody sees the potential, but I think we always wish we can go faster or we can be more efficient. That having program which may require less funds, less resources. I think investors, it's not only today. It's not only about gene therapy or gene editing. It's about the biotechnology market, which is a little bit, which is very difficult, and I don't think there is any specific area of the biotech market which has been privileged, compared to the others. It has been said once again earlier, I think investors want company with a good business model. They want company in control of their technology. They ideally want company who's going to develop drugs for a large prevalent disease. And today, the pricing, the pricing and the reimbursement scheme is still a little bit undefined. So it's going to play, the first editing technology, which will be approved and priced, will be very important to define the rest of the future of the field.
And does it follow, in many ways, the precedent set by other gene therapy, starting off in small rare diseases with small populations, perhaps relatively easy to show and even sell benefit, and then expanding to larger indications in future? Is that part of the value equation that we see now that the investors are going for? Maybe Albert?
Yeah, I think as they look at that, you always want to try to go to a bigger population that you can. I think where gene editing is right now, to the truest definition, going in and modifying DNA at one site. I think it's almost like that's why you see it in monogenic disorders right now, because you have to know exactly what part of the genome is contributing at a big effect size to the disease that you're trying to treat. And that's a great place to start. As we understand a little bit more about larger indications, maybe larger monogenic indications, such, sickle cell is a great example, or even beyond that, once we understand how those genes play a role, then you might see gene editing moving into those areas. But I think it's a perfect spot for the application of the technology right now, just because human genetics has really taught us that is the gene that's contributing to the phenotype, and you have a lot more insight into what you want to do, either turn it off or turn it on.
Yep. Well, I think staying on the theme of the technology, Mark, do you see, is that how you see your progress? You've got quite a big portfolio in Editas.
Yeah, certainly I think there's an element of, picking monogenic disorders where you can control some of the variables, that it's a genetically defined disease and you're really testing the technology as opposed to testing the disease biology as well. That said, there are companies within the gene editing space that are swinging for the fences and they're going after large, for example, cardiovascular disorders. I think from a technology perspective, it's eminently doable, and some of the early data has looked extremely promising. I think the question or the debate will then go to commercialization and reimbursement, because these are large, million-plus patients in some of these indications.
Okay. Devyn, any comments about where it's going with indications?
Yeah, I think one of the things I think that we try and do is follow the biology. I think that's why I think that as Albert said, many of us are starting in rare diseases because the biology is relatively clear. It's straightforward. You know what the mutation is, and you know what the biological output looks like, whether it's in something like a TTR, like Rentelli is, or other indications. I think that's very nice. I think as you move to the larger indications, the challenge is, in a lot of cases outside of, say, PCSK9 or a few areas like that, the biology is very complicated. And I think we don't yet have a broad handle on the underlying genetics, because most of these diseases, as we get into the hood, it's probably going to look a lot like cancer, where Alzheimer's is probably not one disease. It's probably 55 small diseases, each having different genetic drivers. And I think until we really continue to learn that biology, I think the big diseases, we need to follow the biology. And I think some of them, it's emerging, but some of them it's not. So I think it's something we've got to continue to work on. I think as an industry, academia is working on it, but it's exciting to see, and it's going to come in time. And I think as folks have said, the editing technology is there to do what you need to do to the cells. Now you need to get it there, and you also need to make sure you've got the biology you're following.
Okay, and I think it's unusual that almost all this panel is involved in vivo genome editing. I'm quite interested to see, say Mark, you've got in vivo and ex vivo, haven't you? Yep. How do you see the progression from one to the other? Is it a simple transition? What are the main features that differ?
Well, certainly with our in vivo editing, which is for an inherited retinal disorder, I think that's a good compromise over having complexity of in vivo editing, meaning you're exposing multiple cell types and potentially more than one tissue to the editing machinery. You're able to deliver in a very exquisite way, and sort of in combination with cell type specific promoters limit the cell type that gets edited. I think as we move to, say, systemically administered therapies, controlling the editing and controlling the delivery is going to be key. I also think there's probably, one example I particularly have in mind is, ultimately for hematological disorders such as sickle cell disease, in vivo editing of the bone marrow would be the perfect solution, so you can bypass the difficulties with conditioning and so on. But in that scenario, you're editing a complex cellular environment, and you're also having to be very cognizant of what happens to those cells once they've been edited, particularly if it's a stem cell. So I think there's a lot of complexity there, but ultimately we feel that's the direction that we need to go in. And so the question then becomes, okay, well, what technology do you have to develop to be able to control that process as well as you would in an ex vivo setting, such as an autologous CD34 therapy?
Okay, Fred?
No, I think what is great in this case is that we have the ex vivo example, which is a little bit more controlled, as Mark said. But interestingly, I'm always wondering if all the in vivo companies have learned as much as they could from the ex vivo experience. But I believe that, and it has been said, first we will have to follow the biology, and delivery, is something we need to continue to think about, because even if many progress have been done and many companies are working on that, I think we continue to need a more precise and very beautiful delivery in some ways.
Albert?
Yeah, not much more to add, but I do think as you move to in vivo, so we did not have an ex vivo, we were in vivo right from the beginning. And it's really trying to understand from a multitude of components. There are really no preclinical models that you can use to predict what you might see clinically. And that's a big challenge, I think, for all of us. We have an editing program that's in vivo that's very specific to a human cell. And so how can you test that early? You can use a surrogate to go into a mouse or a non-human primate, but you really want to understand the human. And then the other component is, from a delivery perspective, we're using AAV. There are other approaches to deliver. You also have to think about, when you do the systemic aspect of it, not only are you trying to measure what you want that genome to look like that's been edited, but then also how the body's going to respond to what you're using to deliver the genome editing machinery with as well. And so it's a combination and a complexity that I think we're starting to solve with respect to whether it's immune system or other aspects. But all of those components, I think you have to take into account as you go into from an in vivo perspective. And then the last thing I'll add is, we as a company are focusing on the liver. And so we can take advantage of that because using our delivery approach, we can get to the liver at high efficiencies. But then you have to engineer what you want to edit so that if it does go into other cells, you want to try to keep that expression in the liver itself. And so there's some other components that take into consideration as you go in vivo that you need to look into.
Very good. Devyn, I should say. Do you think our technologies are good enough, or the editing can be improved in terms of efficiency or specificity, or is it good enough?
So I think it comes down to what are you trying to solve in the disease. I think the editing technology is clearly good enough for certain indications, for certain diseases. I think that's been demonstrated if you look at some of the clinical data that others have already shown in editing. Are we to the point where we can cure every disease out there? No. I think there does need to be improvements, I think, in the efficiency of some of these next generation approaches where you're coupling a nuclease to some kind of effector. I think there has to be clearly improvements in delivery outside the liver. The liver and the eye and maybe one or two other tissues utilize either LNPs or AAV. You can get there, but there's still work to be done on delivery too. And so I think on the in vivo side, I characterize it that way. Ex vivo, I think the technology to do the editing you need to do for cellular therapies, I think is in great shape. I think there's a lot of approaches. You're seeing a lot of companies doing work there. So I think there it is in great shape. We are not doing ex vivo cell therapy because I've done it and it's really hard. And it's hard to do that and hard to do in vivo in the same company, at least in my experience. So we're focusing on in vivo, partnering in the ex vivo space to try and simplify the challenges we have to solve in one company.
Okay. Mark, where would you want to take the technology in terms of improving what it is now?
I mean, I think the sort of purest use of gene editing technology is actually in the iPSC platform that we use. I think, right now our first product has a double knock-in, a double knockout. I think the excitement here is you can really endow, and I should say we're editing the iPSCs and then differentiating them to iNK cells. The beauty is that you can endow those iNK cells with the properties that really make them much more superior at killing tumor cells. And that can be persistence, it can be homing, it can be resistance to the tumor microenvironment. So in a very specific way, you can tailor those cells for the exact use that you want to provide them for. And I think that has almost endless possibilities. I think what will limit us ultimately will be as we modify more and more cellular pathways, then the pluripotency or the lineage specificity or control of the differentiation will become more difficult. But in principle, we can, whether it's iNK or T or B, any of those types of cells, we can pretty much edit them in a way that produces a single clone that's differentiated to a homogeneous allogeneic population that will do exactly what we want it to.
Okay. Fred, would you like to improve the technology?
Sure. But maybe it might be because my English is not perfect, but I don't really like the good enough. I think all the companies are doing their best to develop the best possible therapy with the current technology, and some decisions are taken by executives and boards and regulatory authorities to help some patients in need, which in most of the cases do not have alternative treatments. And I think we need to continue to make it better. It's already very good, but the science, which is exploding these days, allows us to continue to improve our technology days after days, and all the learnings also from the different trials should allow us to do it better. But I do believe that what has been done to date is already pretty amazing.
Fred, then, what would you like to improve? And also, is there a place for, you've got homologous recombinations, you've got obviously CRISPR, zinc fingers, base editing. Is one of those going to take over, or is there going to be space for all of those?
Yeah. So in terms of, I'll start with the first question first,
okay.
about improving the technology. I agree with Fred and others that we are bringing forward the technologies for the diseases that we're moving forward, and they're fascinating, and it couldn't be a better time to be in the field to bring these forward. But science continues to advance. There are always things that we need to be aware of that we can implement in. Can we improve this or that, and coupling that together? So I do think that we really, it's upon all of us as companies to learn from what we're currently looking at in the clinic, what we're currently looking at preclinically, as well as what our academic colleagues are bringing forward, to see how we can couple that together to make better therapy. So that's the best part about being in this, is you're continuously looking for something that you can innovate and make better.
Maybe one other comment on making things better. I think it's easy to get carried away sometimes with what you could do, but I think ultimately when you think about manufacturing, you have to try to make things as simple as possible. And so making some of the editing technology simple from a delivery and a manufacturing perspective is going to be important.
And you mentioned delivery before. Have you got any great ideas about how to deliver to new tissues, to the brain?
No.
Anyone?
I wish I did. No. Well, right now we use a combination of AAV electroporation for ex vivo therapies and then lipid nanoparticles. Obviously, there's a lot of effort in the field as a whole in developing new delivery technologies, whether it's modified AAVs that have better brain tropism, whether it's decorated LNP, all of that's going on. I think that's going to continue to be a challenge, but I think as we've seen in the field, progress has been almost exponential in terms of editing, and I'm hopeful that someone's going to think of a good idea for delivery to, say, brain, or I'm a neuroscientist by training, there are a lot of very important neurological disorders that are awaiting therapies, and the delivery is really the problem.
No, I'm sure Mark has many more ideas than what he claims, but no, there are many. We are making many progress. We need to test them, as Albert said. Unfortunately, there is no great model to test the delivery, and so it's going to take some time. But I mean, if you look at progress which has been done in any kind of vectors, now there are new viruses which has been identified and could be useful in in gene therapy, and also the progress which has been made in mRNA, but through the delivery by LNPs. I think there is a great hope for much better delivery, but it's going to take time, I think.
Okay.
I think Albert has another question he didn't answer to.
Oh, you throw me on the head. I guess the second part of that question was, would any of these technologies take over? And I think you're hearing the answer to that all of these technologies may be fit for purpose for different areas that they're working on. A lot of it can come down to delivery. I think as we think about, are there ways to cross the blood-brain barrier? There are a variety of approaches. One of the things we're working on is trying to engineer our capsids from an AAV perspective to get across the blood-brain barrier. But then you can ask the question, what do you want to do and edit when you're on the other side of the blood-brain barrier? And that's where some of these technologies still, I think it's too early to say one's going to take over. They're all so unique in what they can apply depending on the disease that you're trying to target.
And I'm just talking about the rare diseases, sorry. There are about 7,000 of them. I forgot exactly how many of them as a treatment, but maybe less than 200. And a handful of them are through gene therapy or potentially gene editing. I think I don't see one technology taking over everything. I do believe that every disease will mandate the technology it requires in some ways.
Yeah, I would agree with that. I think there's diseases where a cut and kill approach is appropriate. You knock something out or down. There's diseases where you need to change an individual base or a series of bases to cure the disease. There's the ones where you need to actually impact regulatory regions. So it really comes down to, I think, that underlying biology. And the reality is there's going to be a lot of different ways we've got to skin this cat. And it's not going to be a one-size-fits-all in any way, shape, or form.
Okay. Whole genome sequencing, and next generation sequencing. Transformative, a blessing, or sometimes a curse because you find things you don't want to know. Regulators are obviously now quite demanding on this subject, especially the FDA. How do you deal with that and see it? And have I got it right? Let's start again with you, Devyn.
So, I will say the developments in sequencing are just phenomenal. Like in our company, we can sequence many, many human genomes in the course of a week, and which generates a tremendous amount of data, which means you then have to have the computational ability to actually figure out what the world is telling you. But also importantly, you've got to make sure you're testing and looking at what you need to be looking at, from an off-target perspective, from an on-target perspective. And so I think it is greatly enabling. I think it's fundamentally changed the way we think about doing these experiments. But we also have to make sure we've got the right infrastructure to be able to deal with that data, as well as do the right experiments so we're testing actually what we need to on the on-targets and off-targets. So that's how we think about it from that perspective. But it is awfully amazing how much, I think of grad school doing the Sanger sequencing on the stupid radioactive gels.
I'd agree with everything that Devyn said. I think the technologies that we have now are incredible. I think good or bad, all data is important and informative, and you just have to have the infrastructure to deal with it.
Albert?
I concur. I think where we've come with sequencing technology is it's unbelievable. It allows you to do things that we could never even dream about before. I think one of the applications is now the long read sequencing too, that you can do very deep sequencing on a single molecule for a long way. And that for us really helps because sometimes we'll have a very long homology arm, and we want to make sure that if we integrate, we go all the way through that and then well into the genome so that there's no question that's not a PCR artifact or something like that. I think the other thing that we're pushing the envelope with some of the deep sequencing and the whole genome sequencing, though, If you're looking for a very rare event, say it's an off-target that's very rare, it's almost like we're limited based on the starting material, so how much DNA from a test model that we can look at to see, because the sequencing will give you that data, but you're going to go beyond what, the starting DNA that you have. So if you have 10,000 molecules of DNA and you're looking for something at 1 in 100,000, you don't have enough starting material to even assess that.
To go back to your earlier comment about the FDA guidance, I mean, they will continue to ask for more and more sensitive techniques. Sometimes, the best we can do is say, oh, it's less than 0.1% or less than 0.05%. That's maybe acceptable now, but it won't be acceptable in the future.
Albert, you're going to disagree, aren't you?
No, I think that's, that's
sorry.
the problem. That's the challenge. So you can set it where you have that limit of detection that's there. And so they will continue. And I think the sequencing technology puts us in that position to continue to go down that road.
I think
Fred,
the two other pieces I think are important is one is being able to know the limits of what is relevant. Because at some point, if it's a 1 in a billion event, but yet you're having 100 double-strand breaks in every cell in your body every single day, at some point, it becomes in the realm of silliness, right? You're like, where is that line, I think? And I think that's where we need to continue to partner with the agency to figure out where's that appropriate line where you're moving beyond the realm of reality. And I think computationally being able to predict off targets is also an important piece of it, because that's another way it utilizes deep computational approaches. You can really be much more predictive of what you may see.
Okay, Fred, surely you'll disagree.
No, actually, I'm noticing I'm the only one without a Ph.D. after my name, so I don't know if I have authorities to comment on this question, but...
That makes at least two of us.
But no, more seriously, I agree with what has been said. I mean, this new science is maybe I forgot to tell you I have a Ph.D. in common sense, actually, but no, seriously, I mean, these new technologies help us to better understand what is happening at the DNA level, at the RNA level, and I agree with Devyn. And Mark and Albert, actually, it's a great tool. It's going to be about how can we use it to understand the relevance of the tool and not pushing the envelope too much, because otherwise it could make the drug development process a little bit difficult.
Okay. I'll give notice. I'll ask for questions from the audience after one more question from this, so get ready. What do you see, therefore, as the rate-limiting technology gaps in the field at the moment? What could be improved? The one that would spring to my mind would be understanding hazards of off-targets when you do see them. The experimental approaches are quite cumbersome, and the theoretical approaches are inadequate. Let's go down the line again. Devyn?
I would say we need more patients treated so we can learn more.
Okay.
Honestly, I think that's probably the biggest hurdle that any technology has, right? A novel technology is you prove the safety in animals, and then you prove it's safe in humans, and then you just we've got to treat a lot of people to and hopefully provide significant benefits. But we also learn a lot about how how does the biology translate? How is the translatability from an NHP to a human? Like, there's so much to learn. We're still really early. So I would say that's the area, we need time and efforts to really optimize that.
Good answer. Not really a technology gap, but Mark.
So I think we talked about this a little earlier. I think it's perspective, meaning I don't think theoretical concern should get in the way of developing a therapy that could be beneficial for patients. And I think we have to take a step back and look at other technologies and other modalities that are approved and also had a theoretical risk, but that has not manifested into something that would prevent that therapy being on the market and people benefiting from it.
Okay, thank you. Albert?
I think one of the biggest technology gaps that we're all facing, we've talked a little bit about it here today, is delivery. So we can get to certain organs we can do direct delivery or even systemic, you can get to some. But there are areas of the body that we just can't get to currently with some of the technologies. There's a lot of work being done on that, but getting the actual editing technology to the right cell is still a pretty big gap to allow us to go into beyond disease areas that are currently there.
It's difficult to be the last one here, but I would agree with Al. I would combine Albert and Devyn. I would say it's delivery, but it's also how can we assess the validity of the delivery system in humans before we go to humans, you know. And I hope the technology will help us to have better translational tools. Would it be real? I mean, like tangible tools or AI or things like that, which will help us to better understand how a new vector can be more efficient at targeting exactly this, doing exactly what we would like to do and not doing something we don't want to do.
Okay, a fair amount of consensus there. So let's see if there's any questions from the audience. And if you do wave, somebody from the ARM would bring you a microphone. Has anyone got any questions for this panel?
This one over there.
Yes.
Hi, and apologies if this was addressed earlier because I missed the start of the presentation, but I'm sort of curious, from where each of you are now, if you only get to invest in one specific area and you get the choice between further developing your editing engines or further developing your understanding of targets to go after in the biology, which do you choose? Targets or the editing system itself?
I think everyone can answer that, and I think we should start with Fred this time.
It's a good question. I think the editing technology is where I would start, and I cannot see you, I'm sorry, but because it's where the safety concern could come one day. And again, understanding how efficient, how precise your editing technology is would help a lot the product development, I think.
Okay, Albert?
I would lean in the same direction to invest in the editing technology, and really for two reasons. One, that's the closest that you're going to get in order to get something to patients that could potentially get some of the learnings that we've been talking about to treat some of that disease. I think understanding target biology is immensely important, but it also can take a lot of investment over a long period of time. And I think that's something that's an area for a great partnership between maybe university academic as well as then trying to think about the editing technology. So from an industry perspective, I would focus on the editing technology itself.
Mark?
Yeah, I would take that into in vivo editing. We talked about that a little bit, and particularly targeted integration or gene addition. I think that's an area that currently there is a lot of effort, twin prime, paste, recombinase, serum recombinases, transposers. That opens up a lot of the target space that's currently accessible by some technologies, but is not the most convenient and not most compliant for patients. And so I would focus on that area.
Devyn?
I'll make it a four on the editing side, because I think the biology, there's a lot of good biology out there for a large number of indications today that I think we can focus on and continue to learn. But I think there's an opportunity to really expand what we do in the editing space. As Mark said, if we get to the point where you can drop in large chunks of DNA, you're not going to need 85 base editing programs to cure cystic fibrosis, right? You need one. And so I think moving towards that, I think, now there's a lot of hurdles to get there, but someday it will happen. And I think making sure that we lay the groundwork to get there is important. I think that's what we're all, we're building that foundation today.
Thank you. Thank you for the nice question. Some more questions, please. Yes, there's one over there.
There was brief mention about the payers. If you had a chance to ask the payers what they would like you to focus on in the next 3 to 5 years, what would their answers be?
Let's start with Fred again.
Good call.
So we haven't done a lot of work with payers, but we studied a lot what other gene therapy companies have done, and what has been also published by other groups. I think that the challenge of the one and done and of combining a one and done and an expensive therapy. I think, 25 years ago, some people believed that we could develop a business model around rare diseases, and they proved it. I think now it's time for other people to think about a very creative reimbursement scheme to be able to allow patients to receive one injection, which might, or one treatment, which might work for the rest of their life, and for payers to agree to be there that day and to cover the rest of the life of these patients. But I think the gene therapy companies have done a pretty good job to date, and I hope that with editing we will be able to contribute to this reimbursement, to this payer environment in a good way for the patients and for society.
Okay. Okay, Albert.
We haven't done a lot of work with the payers, but I do think that the companies that are at that, BLA close to the commercialization are laying the path pretty well for that. And I think what the payers will want is what's the value proposition for them. So are we bringing something that treats and brings significant unmet medical need and brings some therapy to that also would save in the long run some of the aspects that the payers would be expected to pay out in the absence of a therapy. And I think that's where a lot of the companies that are at the commercialization stage now are really paving the path for us, that maybe companies that are following along that way.
Mark?
I think for the rare disease area, this should get worked out pretty quickly, because ultimately it won't be an issue of money, I don't think, based on the number of patients. I think the transition to treating large patient populations is going to be an interesting one. But even there, the precedent, or at least a benchmark, might be set. If the ASI anti-A beta monoclonal antibody gets approved for Alzheimer's disease, that's huge indication. And so there are going to be a lot of payers that are going to be wrestling with how you deal with those types of situations. And hopefully, I was going to say if, but when we get there for gene editing, there'll be at least some process in place, or at least some conversation that's already been had.
I think it's something I'd love to see, which anytime you have a space that's extremely well established, not particularly innovative and flexible and responsive, it's an opportunity for new upstarts to come in and figure out new, different approaches to innovating. And so I think I'd love to see this. I think you're starting to see hints of this, where someone's going to figure out how to innovate in this space on the payer side and be wildly successful and completely upend the way we're doing things. Because, trying to fit the current approach on how we think about reimbursement into into curative one and done therapies, it's not even a square peg round hole. It's so, they're on different planets. And so something has got to give somewhere. And I think we will start to see that. I think even, some of the leaders go out, Vertex, CRISPR, if that's, that will get approved, there's going to have to be different thinking applied, because we just cannot apply these same models, because it's going to limit access to patients. It's going to be a mess. And so I think I'm really interested to see what sort of crazy, wild, innovative startups come out in this space that can change the whole dynamic.
Thank you. Time for at least one more question. Yes, there's one here.
Hi, great panel. I'm just curious, there's so many programs right now, and there's a tendency in our industry to crowd into certain indications, like lots of programs in hemophilia A or B, Duchenne muscular dystrophy, sickle cell thalassemia, and that's with version 1.0 approaches, right? There's gene therapy and some gene editing now. And so now for new innovators who are coming and you're, indication selection, you're dealing with a situation when there's a diminishing pool of patients because they've been perfectly or imperfectly cured, and there's, a bunch of people ahead of you. But that's where the biology is very well understood. Devyn, to your point earlier, it's like we understand that biology, that's where we know that if we do X, we can achieve Y. But now the standard is going to go higher for everybody new starting. So I really, with every new innovator, whether it's in gene editing or any other modality, I really wonder where there's a tension between we really understand the disease biology, and that's the logical place to go, and investors seem to be willing to continue to fund company advancing product like number 10 against that same indication. But you may have to, go into things where the disease biology isn't as well understood, because otherwise you're chasing something that of higher standards, diminishing returns. So how do you guys, as companies sitting on great technology and thinking about building portfolios, think about that problem, that trade-off between really well understood biology but diminishing returns and higher bar that you have to achieve to be differentiated, versus going into brand new areas where you're taking both a biology risk and an editing or technology risk? I'm really curious about that because I think that's something that's facing the entire field right now for every technology.
Okay, thank you for that. Devyn?
Right. It's a really important question for us. I think that the way we've thought about it at Arbor is for your lead programs, it makes sense to minimize all the risk around it. Because if you have so much risk associated, you've got biology risk, you've got, call it API risk on your editor, and you've got delivery risk, and you fail in the clinic, you are never going to be able to deconvolute what went wrong. But if you take a very measured approach with your lead indications, focus, we're focused in the liver where we know we can deliver via an LNP, we know there's diseases with their clinical and regulatory approaches, and then it gets to your fundamental question, what can we do that the other approaches in that disease cannot do today? How can we differentiate clinically? Then you've got sort of a strategy you can grasp, and with success there, then you can actually flip to actually take a little risk somewhere else, whether it's on delivery or biology risk, because then you can still have that ability to deconvolute what the challenges are. So at least that's how I think about it from that perspective. But there's lots of different approaches, but it is a really important question for all of us.
Who else would like to comment? Looks like Fred.
Yes, I mean, that's a question which we are all facing every day, not only gene editing, not only gene therapy. The way I think about it, it's about the unmet need. And maybe to come back to what has been said at the beginning of the panel, there are some patient populations which are amendable for a gene addition program, when you target the liver, for example, you go to adults, potentially we see very good data, but in hemophilia, just to pick an example, but you cannot treat young patients with the same technology, and now we're going to have to decide when an hemophiliac patient needs to be treated. Is it when it turns to 18 or when his liver is more stable, or should it be at the very beginning of its life to potentially reduce the burden of the disease over years? That's more the way I think we have been thinking. We did not develop anything in hemophilia. We've decided to go to a rare disease where nobody was interested, at the time we did it. I think it was a good idea, but it was an idea where the biology was a little bit less clear than in other diseases. So we increased in a significant manner the biology risk. So it's a balance in between. There's two things for me. But.
Okay, we're almost out of time. Quick final remarks. Anyone who's got visions for the future of gene editing, genome editing? Albert?
I think just in terms of where we are with the technology as we go forward, that vision that, as you understand what's causing disease, there are going to be applications that you can go in and try to treat and ultimately cure them. And I think that's what drives all of us, at least at Homology, forward. So.
Okay, 20 seconds each now, Mark.
Again, back to in vivo, well, two things, in vivo editing, I think knock-in technologies, I think that it won't be too long before that gets perfected and the efficiency is a lot better. And then the multiplex cellular editing.
I'd see, you're born, your DNA is sequenced, they'll be like, okay, there are 18 things we need to fix, and you get them fixed. Like that, that's where it's gonna go someday now. Probably not my lifetime, but eventually it's gonna be there.
Fred, last word.
That's a great vision.
Yes.
I would say, I hope that all this experience, all these companies will be able to accelerate the process of drug development and reduce the cost, the scale of it, and potentially develop more and more personalized medicine.
Okay, on time. Thank you, panel. Great discussions, great contributions from you.